Good evening, everyone. Good evening to all the students. Please confirm whether the audio and the video is working fine or not. Let others also join in and then we'll start off with our class. Yes, just confirm whether the audio and the video is clear or not. Then we'll start off with our one more free marathon class, right? We'll be discussing about previous year questions of June 2023. Okay, we'll be discussing these questions. So let others also join, then we'll start off with our class. Okay. Good evening, Akansha. Good evening to other students as well. I guess there's some technical glitch. Students are not able to join. Okay, yeah, now it's working. Okay, so let's begin with our class now. So talking about before starting the video, if you're new to Botany Insider, make sure to hit the subscribe button. In addition, we have our own application. You can download our app from the iOS as well as from the Play Store. In addition to that, we have launched a very new course today itself, which is a PYQ plus a revision course in which you'll be getting one detailed revision of the entire course. In addition to that, we'll be having previous year questions discussed in the best detailed manner possible. Course will start from 18th of September and the enrollments for the same have begun today. The link for the same is in the description box. You can check out the description box, get yourself enrolled, and for the initial 5 days, the pricing for the same is 999 and it will increase after 5 days. Okay. So if you're interested in enrolling in this particular course, you can download our application and get your slots booked for the same. It's a complete new approached course which we have designed specifically for the students who are appearing in the or who will be appearing in the December 2023 with detailed previous year questions. You'll be having a personal CBT practice portal. This is a very unique feature, very new feature which you all can utilize. Then revision classes for all the units will be there. We'll be having weekly tests, hard copy PYQ pointers will be delivered at your doorstep. Notes, soft copy will be provided. Personalized attention will be given and doubt sessions will be solved. Okay. Everything in just 999 and it's a one-time payment. You need not to pay any other amount. Okay. So this is about it. Let's begin with the class. This is the very first question for you all. Everyone, give it a try to the question. Question is, which of the following is the communicating junction between or which links the two adjacent cells in a plant, which is helping in the small molecules to move from one of the side to the other side, okay, while it blocks the most of the large material molecules. Okay, it's a morning shift part B question, just a revision for you all. Give it a try to the question, everyone. 15 of you are there in the class, and I want all of you to participate. Okay, give it a try to the question, everyone. Very good, Kanika. Very good, Himani. Everyone participate. Very good, Shivangi. Make sure that you join in our free classes on daily basis. You learn a lot over here, okay? And for the previous year questions specifically, we have started off a course and it will help you out for sure, okay? Very simple question. The question is, you need to find out the name of the communicating or the linking junction which is helping in the two cells to move, like which is linking the two plant cells adjacent to each other and helping in the small molecule movement. Simple answer. Answer is option number C. Very, very simple question. Excellent. All of you got the question correct. Okay. Remember, this same concept, exact same question concept have been asked so many times in the examination. We'll be discussing a number of previous year questions in our main course, that is the previous year and the revision course, where you'll get to understand that how or in what format are the previous years repeated. Okay. Next question for you all. Which of the following statement regarding the PEP case is incorrect? Okay. You need to write down. You need to find out the answer for which of the following is, like, which statement regarding PEP case is incorrect. Give it a try to the question, everyone. Simple question. Very good, very good. Give it a try to the question, everyone. Read the question carefully and then mark the answer for the same. It is asking you related to PEP case. Mark down the answer, everyone. Let's look into the answer now, right? Simple question. Okay. Let's look into the answer. Answer is option number 1. Very good. Some of you got the answer correct. Some of you got the answer incorrect, but it's okay. During the practice time, it's like, if you make mistake, make sure that you're learning from the same. Okay. You need to find out the incorrect statement regarding PEP case. So during the day, C4 PEP case is inactive, whereas CAM PEP case is active. No, it's the other way around. Try to recall what did you study in the case of your CAM pathway and your C4 pathway. When we look into the photosynthesis, during the photosynthesis, the normal pathway is what? During the photosynthesis, the normal pathway is the light reaction, which is via your Z scheme. If the or if the plant requires some additional ATP, it undergoes cyclic photophosphorylation, right? And then you have the dark reaction. During the dark reaction, you have the C3 cycle operating. But every plant cannot undergo, like every plant have different conditions. So it need to safeguard itself. For the matter of safeguarding, what is required? There are three different types, like two different type of cycles. That is, one is the C4 one is the CAM one. Why does the cell require, or why does the plant need to operate via C4 pathway or the CAM pathway? Reason being, there's a wasteful process known as C2 cycle, which is photorespiration. Why does photorespiration occur? Reason being, you have Rubisco enzyme. This Rubisco enzyme have equal affinity for oxygen and carbon dioxide. If there is higher concentration of carbon dioxide, the Rubisco will work as a carboxylase. If Rubisco has low, like higher concentration of oxygen, it will work as an oxygenase. So, regardless of, like, both of them have the same active site for the binding. So, if higher concentration of oxygen is there, your Rubisco starts to work as what? The Rubisco starts to work as an oxygenase. Because it works as an oxygenase, it initiates a process known as C2 pathway, that is photorespiration. To avoid photorespiration, because in this there is unfixing of carbon dioxide going on. The fixed carbon dioxide gets unfixed, and at the end, C3 is operating in. Okay, I'm not going into the depth of the topic, just giving you the entire interlinking. So, for avoiding this C2 cycle, you have C4 pathway and the CAM pathway. C4 pathway is when you have a like the complete same thing will be there. The only difference is the changing in the positioning of your Rubisco, so that the Rubisco have higher concentration of your carbon dioxide, which is via Kranz anatomy and which is via dimorphic chloroplast, right? Then you have CAM pathway. So CAM pathway operates in those plants which are majorly the ones where there is lesser amount of oxygen being, lesser amount of water being present. Specifically, example succulents, where less water is there, so there need to be preservation of water. For that, what do we require? For that, the photoactive stomata need is required, right? The stomata is active during the night time and not during the daytime. So in case of your daytime, tepcase is active in case of your C4 and inactive in case of the other one. So it's like other way round for the same. Okay. Make sure whenever you are practicing out a question, whenever you are practicing out any concept, you are correlating the entire thing and then you are like marking down the answer, because that makes you revise and recall the concept as well. Okay, so answer for this is option number 1. I hope it's clear. Very simple question. It's all about how strongly you revise. The more strong your revision is, the better you'll be able to perform in the exam, right? Then you have the next question. So this particular question is related to your protein digestion. Very interesting, very easy question. If you remember about the various enzymes related to the same, related to the protein digestion concept, you'll be able to mark down the answer. Okay, give it a try to the question, everyone. And remember one more thing, if you are getting some answers incorrect initially, make sure that you're not fearing from that. Okay, you're not like having any kind of burden on your mind that you're not able to solve it. Try to improve as much as you can during the practice session. Okay, give it a try to this. Good evening, Nisha. Mark the answer. Give it a try, everyone. First option is chymotrypsin does not generate peptides with a C terminal. Second, trypsin generates peptides with C terminal basic amino acid. Then you have carboxypeptidase B act on aromatic one. Carboxypeptidase A employs zinc ions for hydrolysis. Then you have the brush water enterokinase has no polypeptidase attached to it, polysaccharides attached to it. And the last one is the final digestion of the amino acids occur in the intestinal lumen, the brush border, and the cytoplasm of the mucosal cells. Which of the following option represents the correct statements? Give it a try to the question, everyone. Done? Okay, let's look into the answer now. The answer for this particular question. Is it working okay? Like, I guess there is some issue. Hello, hello, hello. Hello, hello, hello. Is it working fine? I guess it is now. Okay, okay. So the question is the following statement are made about the digestion of the proteins and the enzymes that are involved in it. Okay. So first option is chymotrypsin does not generate the peptides with C terminal amino acid, C-terminal neutral amino acids. Okay, if you look into the first one, that is chymotrypsin, it works with your carboxyl N. So carboxypeptidase and aminopeptidase they are exopeptidase that remove the terminal from the terminal residues from C and N, both of the terminals. Okay, so this makes the first statement correct or incorrect? Yes. This makes it correct or incorrect. If it is working with both of the sides, so is it making it correct or incorrect? And it works with what? It works with tyrosine, phenylalanine, and tryptophan. That means not with the neutral one. So A option gets incorrect. If A option gets incorrect, then yes, participate in the class, everyone. It's a lengthy one. You give it a try to this. We'll solve out that question in the different one because it's a very lengthy question. I won't be able to cover up the remaining one there. You quickly mark the answer for this. I want to solve 6 questions. Why is it flicking on my end? Is it working the same way? Like, I guess there is some technical issue. On my end, the screen is flicking. Hello, hello, hello. I guess there is some technical issue. The screen is like not stable on my end, but other things are working fine. You give it a try to this question. Those of you who are like able to see the screen properly, you give it a try to the question. Hello, mark the answer for the question, everyone. Is the screen flicking on others end also? Yeah, screen is not stable on my end as well. But still, like, give it a try to this particular question, everyone. Let's do one thing. You solve it out and I'll continue tomorrow because I guess there is some issue on the YouTube side itself because from my end, everything is working fine. Give it a try to this, everyone. Solve it out. Now it's stable. Okay, perfect. Okay, everyone, give it a try. First option is 5 that it is related to the factors that regulate the fidelity of the DNA replication. What do you mean by fidelity? Fidelity means the accuracy, right? The best possible way in which the or the most correct way in which the RNA polymerase works in to add on the new nucleotides, right? So 5 dash to 3 dash exonuclease activity of the replicative DNA polymerase, then imbalanced intracellular concentration, then you have removal of incorrectly incorporated nucleotides by the mismatch repair system. Then you have increased intracellular concentration of the RNTPs, resulting in increased incorporation of RNTPs during the DNA synthesis, which are not easily removed by the polymerase proofreading activity. You need to find out which factors regulate the fidelity of the DNA replication? Simple question, easy question. Give it a try. Done, everyone? Simple question. Let's look into the answer. Yes. The answer for this particular question is option number B. That is B, C, and D statements are correct. C. First one is the 5' to 3' exonuclease activity of the replicative DNA polymerases. Second option is imbalanced intracellular concentration of the 4 DNTPs, then increased intracellular concentration of RNTPs, resulting in increased incorporation of the RNTPs, and hence, like, which do not get removed by your RNA polymerase proofreading. And last is removal of incorrectly paired via the mismatch repair system. So the first option is incorrect. Except for this, the remaining one are the correct one. No, answer should be option number 4, not option number B. Okay. Second cannot be the correct answer. This one is the correct one. Then B is correct, and then you have option number D correct. Removal of this option is incorrect. Okay. Answer should be option number D as far as I remember the concept. Just a second. Answer should be option number 4. Just a second. Okay, sorry, I read it incorrect. It's 5' to 3'. Sorry, the proofreading activity is 3' to 5'. Sorry. First is incorrect. So answer is option number 2 itself. This one is an incorrect statement because 5' to 3' is replicative activity and 3 to 5 is the proofreading activity. That's why the answer will be option number B itself. Correct. The answer for this will be option number second itself. Okay, so remember this particular question. Like, let's keep the class till here because I guess there is some technical glitch which is occurring. So I'll take up the class in the next session. We'll be like, let me check what the technical problem is going on right now. So those of you who wish to join in this PYQ session, you can join in. I have shared the link for the course as well. You can download the app and get yourself enrolled. Okay. So sorry for this technical thing. I'll check it up and we'll have a next class in the like tomorrow or day after tomorrow. We'll have our next class. Okay. Make sure to download the application. Yes, the screen, there is some technical problem. There is some issue today. So let me correct it first. Okay, I'm keeping the class in here.